Good morning and good evening, good afternoon, everyone. Thanks again for coming to this meeting in the final day in this early Sunday morning or Saturday evening. So I added a word exploratory in my title, so I will explain what I mean by this. So I'd like to start by sharing my link with R and bioconductor. So actually I have some picture to show you that was in 2001 or 2002 in Boston when I had some overlap with Robert Gentleman. We had listened to his talk on the first day. So at that time, bioconductor was not born yet, but R was already there. So thanks to Robert's work. As Matt said, I had some contributed some packages to bioconductor. The one, probably the most successful one, is DEG-seq for identifying differentially expressed genes from RNA-seq data. This is actually one of the earliest packages specifically designed for this task after RNA-seq technology became available. And it was interesting to check the feature that bioconductor provided for the summary on the downloads of each month. So I picked up some examples. So that was, the package was released in late 2009. We see the download here. And then it increased dramatically. So that's 2011, 2013, about 500 to 2,000 downloads every month. And it was interesting that there has been many software coming and go, but now somehow the DEG-seq is still alive now. So this is 2020, and this is this month, so October. So we can say that the downloads keep active as almost as the same. So which means that DEG-seq has been also applied on many single cell RNA-seq data. And in recent years, we have also developed a tool that is specifically designed for single cell RNA-seq data. So that's the comparison of two groups of single cells and find the differentially expressed genes. A unique feature of this DE-Syncal package is that it not only reports the differentially expressed genes, but it categorizes the DE genes into three types. I will show it here. So the first type, we call it DES. That means that the status of expression are different. I use this cartoon to explain the idea. So this is one group of cells and the other group of cells. So there are situations that in this group, many or most of the cells are not expressing this gene, but with only a small portion of them express this gene. And in the other group, most of the cells express this gene. Only a small portion of the cells miss this gene. However, for those cells that do express this gene, the expression distribution are approximately the same. But if we take a look at, we compare the two groups, we will find a significant difference between the gene expression. So we call this difference as the status, because the on-off status of the gene has a significant difference in the in these two groups of cells. The other situation is more standard, that we call it DA, meaning that the abundance difference. That is the proportion of expressed, the proportion of cells expressing this gene are approximately the same between the two groups. But for those the cells that express this gene, the distribution has some difference. This one is much lower than this one. And of course, there is a more general version, we call it DEG, that is the mixture of these two effects. We think the biological mechanism behind these three groups are different. So we added this feature by developing some mathematical model behind that. So if you are working on a single cell RNA-seq data, probably you may try to use this DE single to break down the differences of the gene expression. So those are some of the, as Matt mentioned, we have also some other packages, but these two probably are the most popular one. And the topic I will talk about is more machine learning. It's a kind of a experimental work. We are thinking on what is the future direction of machine learning in the context of biological knowledge discovery. We will have to, before telling the story, so everyone is know that we are evidencing great advances of machine learning and artificial intelligence these days, from image recognition, automatic captioning, autonomous driving, medical applications, and AlphaGo, and also these fake videos from this picture and machine translation. And this is my colleagues work at Tsinghua University. So it's kind of autonomous bicycling. So machine learning has been very successful, and not only machine learning, it's all artificial intelligence in general. But if we really look into the details, we realize that the current machine learning is mostly, I call it imitative learning, in the sense that for supervised learning, what we do is actually we want the machine to imitate the training samples, and the training samples were given by humans. So all the tasks have, so for humans are able to perform the tasks, although not necessarily better than the machine. But those tasks are what human beings are able to do. So this is for supervised learning. And for unsupervised learning, I think basically what we are doing is estimating distribution, either is discrete distribution that is clustering, or continuous distribution that is kind of a manifold learning, or find some dimension reduction, things like that. And after this clustering or dimension reduction or manifold identification, we need human involvement to annotate the results guided by given knowledge. So current machine learning, so in this sense, I think it's not good enough for knowledge discovery. So a question came into my mind is that, can we use machine learning to find new knowledge that is really unable to be identified by human beings? So that was my motivation, and I coined this term exploratory machine learning. Say that the existing machine learning are all imitative learning, and we should try to develop methods for exploratory machine learning. So after coining this term, I checked the website to see whether this term already exists, and found that it is a well-established term in education science for human beings. That is, exploratory learning for human beings has 4 principles. That is that learners, that is human students, can and should take control of their own learning. And knowledge is rich and multidimensional. And learners should approach learning tasks in very diverse ways. And it is possible for learning to feel natural and uncoaxed. That is, it does not need to be pushed. So that was the features for human exploratory learning. And the question is, can we do it for the machine? So the later 2 aspects are involved in the motivation. Probably currently we cannot expect the machines to really have a motivation. Then I focus on the first 2, whether we can let the machine to really take control of their own learning and to learn rich and multidimensional knowledge, not only the clustering or some trajectory that's kind of a low order knowledge. So that's my motivation, and we this is ongoing work. We are exploring this field with several trials. The one is that we try to work on the CT image, the medical image for non-small cell lung cancer tumors, and we ask the question whether we can find heterogeneity within the tumor with machine learning. So we know non-small cell lung cancer is the most abundant. It's the form of lung cancer, which is the first cancer of human being. And lung cancer have different genomic reasons, that is the heterogeneity, but we focus more on the so-called intra-tumor heterogeneity. That's within each individual patient, the tumor still have cells with very different genomic background. And this is the major cause of the recurrence of the disease or the drug resistance of the target therapy. So identifying this heterogeneity, intra-tumor heterogeneity, is important. However, if we want to study intra-tumor heterogeneity, we need to do biopsy surgery and do genomic study. And even we do this, it's not necessarily easy because tumor is large and we can only say in biopsy, we can get a few samples from it. We may not be able to really capture all the genomic features. And on the other hand, the CT image have been the standard of for tumor diagnosis. But the application of CT image is basically the detection of the tumor and the location, and also try to identify whether it's how severe it is. So, and people have been using machine learning technology to predict the benign or malignancies of the node from CT image, but people have been not using it for heterogeneity study, either by the clinical, by the clinicians in the practice, or by machine learning. So we ask the question whether it is possible to get more information than the information currently used in the clinical practice. So that is the motivation, and we need to check the mechanism of this CT machine. We know that the CT image actually are not image; they are kind of pseudo image. We use this signal to scan the body, and because different body components have different attenuation coefficients for the signal, we can distinguish the different parts of the body from the signals we received. And we need some mathematical algorithm to reconstruct the image to show it to the doctor. So the original data we collected are actually not directly the visible image. And after converting it to the visible image, we know that the dynamic range of the signals in the CT data are much richer than what human eye can perceive. We have to need to set some window to see different contents in the body. So that from this understanding, we say that what human eyes can perceive from the CT image may be only part of the information in the CT image. Using computers, using machine learning, we may be able to extract more information than that has been used in the clinical practice nowadays. So that was the thinking. And then with this reasoning, we try to develop a method to see whether we can get some information about the intra-tumor heterogeneity. The idea is that we get the nodule of the tumor, and we get the image here, and we want to do the analysis of each pixel, so we are assuming that if there are some heterogeneity within this tumor, it might be reflected by some property of each pixel. But each pixel is only one pixel. There's only one data point. There's not much information unless we put it in the context of its neighbors. So we use a small neighborhood of each pixel and to extract features and do clustering on each tumor. Clustering for all the pixels, for all the small area around each pixel, and do clustering. And we hope that this clustering will show some pattern like this. And if we see this pattern, we hope that the pattern should reflect some intratumor heterogeneity. But there is the issue that because we don't have other data, so if we find some pattern, can we really explain that? So that's the puzzle we need to solve. So now I will go through what we really have done. So we extract the features that are a lot of radiomics features. Actually, we use the existing packages for these features, and we do the clustering. And of course, we meet the problem of how many clusters we want to see. And we explored different choices. Say we use that's the standard k-means clustering, and we tried different number of clusters. And interestingly, we find that when we change the number of clusters, of course the look of the distribution of cluster will change. And we hope that there may be some pattern. And so like this. So this is one example. We get the nodal from the patient and like this. And we do clustering. If we do two clustering, the clustering for two clusters, we find the two clusters are like this. They are one is in the center and the other is around. And if we do like k equals to 3, we get this, and so on and so forth. And so every so when we increase the number of clusters, so this is just a final discrimination of the clusters, but the shape keeps the same. And there is a center core. Seems like there is a center, there is a core in the center of the tumor. So we call this as the uni-core pattern. So this is interesting because after we check the hundreds of examples manually, we find that it's not all like this. And there are situations like this. So these are the number of samples. So in the data we studied, so almost 500 falls more or less into this pattern, and another 500 or more like this. So if we increase the number of clusters, so the pattern changes, but there seems to be more than one core, and the core position basically stayed. And there are some other cases, say like this one, it's more, it's 800 cases. So in those cases, when we increase the number of clusters, there seems to be no pattern, no consistent pattern. So we reason that probably that there are 3 major patterns that we name it as unicore, multicore, and diffuse pattern. And then, yeah, these are the numbers. So we have looked at two data sets. These are the numbers. And then the question is that whether these patterns are informative or whether the information in this pattern are useful. So fortunately, in the data we worked on, they are public data. So in the data, they have the survival information. So we reason that probably if the pattern has any relation with the heterogeneity, and if the heterogeneity has some relation with the severeness of the disease, then we reason it probably has some association with the survival time. And we draw the survival for the two extreme cases. One is the diffuse the pattern, and the other is a unique pattern. So we can see that there is a significant difference between these two groups in terms of survival time. And this is another data set. And so this gives us the confidence that that we think that these patterns reflect that some of the intra-tumor information, and this the intra-tumor heterogeneity, the difference in this intra-tumor heterogeneity among different patients are associated with the disease outcome. And this is yeah, we put the third group in this MK plot, and we say they are between. So somehow we can guess that this unique core is more homogeneous, and which implies that better prognosis, and this diffuse may be more heterogeneous, and which may result in worse prognosis. So that is the story. But all these analyses are done by human beings. So we analyze the patterns by our human eyes and organize them into 3 groups. Then we ask whether we can use machine learning to do this. Of course, this is not knowledge discovery yet, because we kind of human being discover these 3 patterns. But then we can say we train a convolutional neural network to see whether we can let a machine to mimic what we have done. And we use that, we do the prediction of these 3 types. And it was quite successful. We see this accuracy of predicting this heterogeneous pattern was quite good. So this is the first story. The take-home message is that it seems possible to find information on intra-tumor heterogeneity from clustering patterns in the CT data, which has not been utilized in current medical or biological studies. And of course, there could be other factors that are associated with this clustering pattern. So this is still just the start of the study. And to get a better understanding of this topic, probably we need to have data that we have the radiomics data, genomic data, and transcriptomic data for the same patient, same tumor. But this is not that easy nowadays. So it's not, the difficulty is not only that we get the same data for the same patient, but all this data for the same patient, but even if we get the same data, it's hard to align them. For example, if we do biopsy or surgery, it is hard to align the images to the genomic or transcriptome data, but probably this is the direction we should go. And what we have done, we can summarize it in the whole logic can be summarized in this graph. So we are assuming that a tumor may have genomic heterogeneity, and this genomic heterogeneity, that is the difference in the mutations of the tumor cells in the same tumor, will cause the so-called intra-tumor heterogeneity. And such heterogeneity may result in patterns in the CT image. And these patterns, hopefully we can identify them using clustering analysis. And also, on the other hand, this intra-tumor heterogeneity may cause differences in the survival with the same treatment. And our logic is that now we find some association with the clustering pattern and the survival time. Then, because all these are has question marks, so we don't know. But we find association at this end. So we are kind of inferring that the existence of this causal relationship. But this is a kind of a simplified scenario that is that survival time has other causal factors, a lot of other causal factors we didn't consider. And also the patterns in the CT image has also a lot of other factors. So those confounding factors may affect our reasoning, but we hope that with more data and more understanding of this scenario, we probably will get a better understanding of the topic in the near future. So that is the first story I want to share. And the second story is, let me check the time. Okay, yeah. So the second story is about single cell RNA-seq data. So we proposed the idea of initial knowledge discovery. I will explain why I'll call it initial. So we know that almost everyone are working on single cells these days. And if we look at the single cell RNA-seq data workflow, bioinformatics workflow, it's like this. We need to have stringent quality control for the original data, for the raw data, and then we quantify the gene expression and get the original data matrix. And we need a lot of cleaning or filtering or batch effect correction and or selection to clean this original data to get a cleaner data matrix. And then probably we will need to do some dimensionality reduction to get a smaller matrix. And after that, we do taxonomy analysis, trajectory analysis, spatial analysis, network analysis, and cell-cell interaction analysis, so on and so forth. But from the machine learning viewpoint, these are all different aspects of machine learning or data mining. And then we build the so-called human cell atlas with this workflow. And if we look into the details of this workflow in some, say, the most representative work, we will notice that all the processing have heavy dependence on existing knowledge. Like we pick up this great work published a few years ago on Science, and this is a breakthrough work in immunology study using single cells by the broad scientist. So we try to reproduce this work and realize that this is the pipeline they use. A lot of steps. And so the first question is why we should use this pipeline. This is just I think this is the result of trial and error. And if we look deeper into this pipeline, we notice that most of the steps involves heavy human interaction. So heavy interactive choices and selections are needed in these steps, and all those choices and selections are based on human knowledge and human judgment. So that's why we call that this is knowledge discovery, but it's not an initial knowledge discovery because it's started from human knowledge. It's not started from nothing. So then we got the question: can machine learning really reveal complicated biological knowledge without using prior knowledge or human judgment? So that was our curiosity. We want to explore whether it's possible. That again, it falls into this exploratory learning, and we want to see whether this is feasible. So we pick up another data set on the single cell development, the embryonic single cell development by single cell study of the early days of embryonic days 3, 4, 5, 6, and 7. And these are the original data, the work they have done. And these are from day 3, day 4, day 5, we say this the data distribution. And so we reprocessed the data and selected some 500 genes without using any biological knowledge. We just use the highly variant genes. Of course, that some biological knowledge that is those informative genes must have some variation. If the gene is always constant, there's not informative. This is kind of assumption we used. But other than that, we just select 490 genes to start with. This is the t-SNP plot we got. It's similar to that. So it's okay. So we want to ask whether we can study, we can discover something interesting there. Interesting in terms of for the sake of the machine, not for scientists. So scientists already know a lot in here. But the machine does not know anything. We want to see whether machine can find anything interesting. So the key, the basic information here is that the lineages of the cell development. And these are the original, the figure from the original paper. There are 3 known lineages in these early days. One is the pre-lineage, that is before differentiation, and after differentiation on day 5, the cells are differentiated into ICM and TE2 lineages. So we want to ask this question. So suppose we have the data without any knowledge. We only know the data of 5 continuous days, day 3, day 4, day 5, day 6, day 7. And can we use exploratory machine learning to discover the knowledge about this lineage? Because this lineage relation is the simplest and also the fundamental knowledge in these 5 days. So that was our question. And so we tried different strategies, and this is what we end up with. So we got the knowledge, and the philosophy is that so we use unsupervised learning on each day. By the assumption that if there is some lineage, I mean, if the cells in the day contains more than 1 lineage, then we probably we may identify them from this unsupervised learning. And we find if these unsupervised learning results are the true lineage, then we should be able to find associations of the cells in the other day with this cluster. Then we use this unsupervised learning result as a reference and use a supervised learning to predict the classes of the cells of the other day. And then we want to check whether the classification based on this reference day are consistent with the clustering of that target day itself. So again, for that target day, if it contains multiple lineages, then we also do clustering on that day. Although we do not use it as a reference, then using this reference to predict the classes and do clustering on that day in unsupervised, they should be consistent if this clustering and classification revealed the true lineage. So that was the reasoning. And we do it for each day. We use each day as the candidate reference day and make a candidate story. And then we get 5 candidate story. Actually, we can get more. Each story gives the kind of the machine's guess of the developmental process. And then we need to find a criteria to justify which one is reasonable. So this is, we do not want to involve human knowledge to judge which one is right. We want a machine to make its own judgment. So that is and with this, we let the machine to decide some story. And of course, this story has no biological terms. So we use the prior knowledge to annotate this so-called machine discovered story, and we see whether it makes sense. So that is the whole pipeline, and this is the detail of this method. We just use the cells of each day and do clustering, and use this clustering to train a support vector machine to predict the other days, and this will make a story, and we try to just to judge which one is the better one, and then after get the best story, we put the prior knowledge in it to explain or annotate the knowledge. And the first step, of course, again, it is how do we decide the number of clusters? So we first try to use the so-called score for deciding, for choosing the number of clusters, and these are the number of the numbers get the highest score for each day. So we use this 2 and 3 for these days as the K for the K-means clustering. And but we checked the script part of the K-means clustering and find that this there is not a very dramatic change at these points, so which indicates that the number of clusters is not that clear, but somehow we fix this and use that, and this is the result we get. That's we use K equals to 2 for day 3 and 2 for day 4, 3 for day 5, so on and so forth. And with this, we get a cluster result on day 3 cells, and we use that as reference to predict the other day and get and that is we use day 4 cells as a reference and predict the so-called lineages for the other day, and so on and so forth. And these are the number of cells we got for each of them. And then with this, we get 5 stories. They are different. We say these 5 stories. Sorry that you cannot read it, the details, but I will just give an example. For example, for day 3, so day 3, the story, if we translate it in human language, the day 3 is the development starts with 2 lineages on day 3, and one of them disappeared on day 4, and there is only 1 lineage in day 4, and so on. For day 5, day 6, day 7, there is only 1 lineage remain. So of course, with our human knowledge, we know this is not true. But can we let the machine to identify this is not true by itself? So that and so on. Yeah, from human knowledge, we can judge which one is probably the correct answer. But we want the machine to do it by itself. So the idea of let the machine to make the judgment is that we give the assumption that if for the story to be true, the classification and clustering result should be consistent. There is an underlying assumption behind this reasoning, of course. So that is the lineage is the major information for the clustering, and also the cells of the same lineage on different days should share the same features. So this is an assumption. So for this judgment, we define a few scores. One is called Concord score that measures the consistency between the clustering and classification. That is the clustering, and that is the so this is the S is the clustering of day I, and C is the classification of day I based on day R as a reference. So we use this as a concordance of the classification result on day I, and each cell, each day is not equally reliable to be a reference. So we define this reliable score as the average of the score, the Concord score when we use this reference. So the idea is that if we use this reference, if this reference makes all the other days have a poor Concord, then we blame this reference, not blame the other days. So this is really the relay. And then we summarize them together. We call it the adjusted reliability score. And this is kind of weighted reliability with the Concord score with the reliability of each day. And we use this for the story. For each story, we get this score and we can get which one is the highest. So this is for the 5 stories, and we get each score, and this is the summary score. We can see obviously this day 5 has by large it's the best score we get, and then we decided actually the machine decided this is the story to be made. And this story, these are the number of samples of each lineage, and this story tells that the development starts with one lineage, we call it A5 on day 3, and then it's still most of the cells are of this lineage on day 4, and some new 2 new lineages appeared on day 5, and the first lineage disappeared on day 6 and day 7, and that's what they said. It's the story the machine discovered, and now the machine has reported some story. We need to annotate it, or we need to align this machine-made story to the human knowledge. So we check the literature, find the knowledge, and the knowledge of this period of embryo development is that on the first 2 days, on day 3 and day 4, there are only pre-lineage cells. And day 5, there are 3 cell lineages. The pre-lineage is still there. The new lineage that is called TE, and another lineage is called ICM, inner cell mass, which later differentiated into 2 sub-lineages. So, and that's for day 6 to day 7. So we need to align this to the story. This is human being. We need to use, we people need to align this. It's easy to align this pre-lineage with A5 because there is only 1 lineage in day 3. But how can we decide which one is which one on the B5 and BC, which one is TE and which one is ICM? So we try to do the clustering. So this is a human involvement again. So as we know that on day 5, if we look at a raw resolution, there are 3 lineages. But if we look at a final resolution, there should be 4 lineages. Then we let the machine to let k-means to do 3 clusters and 4 clusters, and we see which cluster splits. Of course, maybe both, so all 3, if all 3 clusters split into new clusters, then the logic fails. But fortunately, we succeeded. So we found that this cluster split into 2 clusters when we let k equal to 4. Then we decided that B5 is TE and C5 is ICM, because this C5 can be further differentiated into 2 subclusters. So that is the human reasoning again. So and there is a contradiction in this human knowledge, the pre-knowledge and the machine discovered knowledge. That is, there are 10 cells on day 4. They already be the TE lineage. So these are zoom in, we find that. And then we check whether this is some bug or this is something really new. And we look at the heat map, we see that these 10 cells really have a very different expression pattern with other cells. And we also put the day 3 and day 4 cells into the PCA plot of the day 5 cells. So these PCA, these are the principal components of day 5. But we put the cells of day 4 and day 3, we mapped them into this day 5 plot. And we find that all these, the pink ones here are the day 3 samples, and these the crown ones, or the lighter green, or you may call it, these cells are from the day 4 samples, with exception some of the 10 cells that are kind of outlier in this day 4. They really map to this TE group in the day 5. So we confirmed that this is not a bug. This is a new discovery that the differentiation of the TE cell already started in day 4. It not happened in day 5 as the literature reported. So, but there is something we are not satisfied with the work. So that is so the choice of the cluster numbers is not that automatic. And then we later we went develop another strategy. Actually, that is the reviewer asked us, and we just explored all the possible cases and let the machine to decide. So so we explored k equals to 2 from 2 to 10 for each day, and we get multiple stories for each day. Then we get a lot of stories, all these stories, and we take the vote. Let the machine to judge which story is the best. And we are glad that still the machine gets the top story is a day 4 using 3 clusters as a reference, the same story as we have seen. And so this is the whole method. And we also applied it on the mouse data, but for, unfortunately, this data is very simple and there's no lineage change. So everything is, there is a 3 lineage all through this day 5.25, 5.5, 6.25, and 3.5. So they are closer in the development process. So there's no not much dramatic thing, but we also identified that, I mean, the machine also identified the correct story. And we also applied it on the zebrafish development data, which is much more complicated. It's from 4 hours of the development to 24 hours. The machine found some story. The complication is that when we try to annotate the story with no knowledge, we find that the no knowledge is really massive knowledge. So in the original paper published in this data, they actually find 180 subtypes or 180 cell types in this period. It's a much more complicated developmental story. And this is a much longer time than the 5 days we compared in the human development. This is more than weeks or even months if we map it into the human development time scale. So the machine can do something, but it's not much. It can hard. We cannot expect the machine to really discover this complicated story like this. So the take-home message is that somehow this initial knowledge discovery can do something, but we still need a human involvement at least at the later phase of this study. And but I think this is a quite interesting thing. In recent days, a lot of people are talking about what AI can do for biology. So we are thinking that this exploratory learning should be a future direction that we focus on. It is challenging, but if we do this, then we will really probably get to the new age of the so-called AI biology or things like that. So with this, I will end here. These are the people actually I have mentioned. This I have not mentioned, but shown the names of these people. And these the students I circled in yellow are the ones who worked on the first on the lung cancer CT image project, and the others are on the red are worked on single cell knowledge discovery project. So I will stop here, and we are actually, I'm the executive editor-in-chief of this quantitative biology journal, and we can sponsor, joint sponsor this meeting. So I will stop at this slide. Thanks.
Thanks so much, Dr. Zhang, for a really clear presentation. Questions? We have one in the chat box from Chris. For the second part of the talk, Chris asks, how do you think your approach would work on cells with oscillating states or stages? For example, naive T cells to activated effector T cells to resting memory T cells in the setting of pathogen infections. And thanks for an inspirational talk, Chris adds as well.
Yeah, thanks for the question. That was a great question. Actually, that is the current is a limitation of the method. So this is quite primal work. So we just want to explore the feasibility of this knowledge discovery. And I think the scenario you asked is similar to this one. So when there are oscillating changes, probably the current strategy may not work. So that is so the current the requirement of the current strategy is that there is one reference day that we see all the possibilities. Why this zebrafish data does not work that well because it's because that according to knowledge, there is no single day that can show up all the possible cell types that have been there in this developmental stage. So I think for the oscillating scenario, probably it's a similar scenario. So we may need to find other strategies. So this is just a beginning of the study. We will be glad to look into this the new challenges.
I have a question while people are thinking of other ones to put in the chat box. So for machine learning research, do you find that you use tools in R to do this, or in Python, or in other software environments? How do you implement your machine learning algorithm?
Yeah, so for complicated machine learning algorithm like deep neural network, we use Python. And but I really probably this is done with bioconductor because this is the k-means and support vector machine are quite standard machine learning tools. I need to check. So yeah, I can't remember the detail.
Any other final questions? We've got about a minute to go. Feel free to pop them in the chat box. Here's one from Julie. Thanks for a great talk. For the first part of the talk, if you take tumor stage into consideration, will heterogeneity still have significant effects on survival?
Can you repeat the question again? So if I check what?
Tumor stage into consideration.
Yeah, that's a good point. But the thing is that the data or public data and the information were limited. I think the tumor stage, they are mostly stage 3 or a kind of a late stage. It's not that there are not a lot of choice for us to do much on the other information. So we know, of course, the other information should get involved in this causal reasoning. Yeah.
I think that might be it then. Well, thanks, Dr. Zhang, for a really excellent presentation to start the morning off. We thank you for your time. It's reassuring to hear that there's still some human interaction required. We won't have papers with just all computers as the authors. They'll still need some human co-authors.
Yeah.
Thanks so much, and thanks for hosting us as well.